Hey guys, it's Landon. I'm gonna make sure I'm live before I start. Let's. This is my first time really doing a video with the StreamYard, so I'm just making sure that everything's going smoothly. Okay. Hey guys, I know it's been a while since I made a video. I do want to start being more active again. Because there's been a while, I wasn't really. It's been a while since I've uploaded any videos or anything. But for this video, I'm going to be discussing something that's not really young Earth evidence, but it is evidence for creation. I'd even say that it's an overlooked and underrated piece of evidence for creation. And what I'm talking about, though, is intelligence in microorganisms. For about 3 years now, I've been pretty vocal with my claim that microorganisms display pretty amazing intelligence and foresight. And as a psychology major, okay. I think my mic messed up. I've been having some technical difficulties with this. I'm not sure exactly what's causing this. But yes, I will pick up where I left off. Um, as a psychology major, it is very interesting to me, and it's a remarkable testament to God's glory, this intelligence. I kind of say it's underrated evidence because it's not really well known. Some creationists may be hesitant to count it as exceptional or breakthrough evidence, and evolutionists usually deny that any such intelligence is present. Intelligence is present. Because I have talked with a few evolutionists before, and they kind of seem like they don't seem like they think it's kind of a silly idea to suggest that something like bacteria or viruses could be intelligent. But I think that these sources and information that I've compiled will make it pretty obvious that they are. But before I start reading from the slideshow, I do want to mention a part of my argument not included in the presentation. I didn't include it because I felt it did feel it felt out of place, I guess you could say. But the argument that I'm making though is in an older scenario, bacteria would be some of the oldest organisms around, especially considering that in an older timeline they appeared only 340 million years after the formation of the planet. Some of the cyanobacteria, alleged to be over 3.3 billion years old, already possess the ability to use photosynthesis, and these ancient ancestors are pretty similar to today's cyanobacteria. My point is that this intricacy and likely intelligence would probably had to have appeared very early on in life's history, and it makes me doubt that natural selection could result in this level of sophistication so early on, especially considering that we just see kind of back and forth variation today. I mean, we don't see like actually gains in information. I mean, I think that when you see my slides and sources, you'll see what I mean and how it's unlikely for something like this to have evolved. But I mean, that's the main part of my argument is that essentially this there doesn't seem to have been enough time for all of this intricacy to have evolved, especially considering how early in Earth's history bacteria appeared. But yeah, I'm going to start the slideshow in a few seconds. I'm making sure I'm not having any more technical difficulties because I had some been having some issues with this. I'm not sure if it's the internet connection or what exactly. Let's see. Okay. But yeah, I have um that was part of my argument though. But the main argument, however, is discussing the intelligence of the ciliates, bacteria, and viruses in detail. So I'll go ahead and start the presentation. Let me share my screen. I'm going to okay. Okay, there we go. Okay, so I'm going to start with the main slideshow now because I have pretty much gone over everything I was going to go over for the opening, especially consider the other argument though about the cyanobacteria and all that stuff. I already covered that, so I'm going to go ahead and start on the slideshow, which is the title of the video, of course, Intelligence of Microorganisms, a Testament to God's Glory. So what exactly is intelligence? There are numerous definitions of intelligence, and these definitions vary greatly. For this presentation, I'll provide two definitions from Merriam-Webster. The first is the ability to learn or understand or to deal with new or trying situations. The second is the ability to apply knowledge to manipulate one's environment, or to think abstractly as measured by objective criteria, such as tests. Microorganisms have no brain and can be mistakenly thought of as primitive and simple. However, through this presentation, you'll see how clever and intricate it can be, showing that God's fingerprints are visible even at the microscopic level. So I'm going to start with ciliates. A little on ciliates. They are a group of protozoans that possess cilia, flagellum-like structures used for mobility. Their diet often consists of bacteria and algae, though some of them do prey on other ciliates. Paramecia are actually an important model organism. They're commonly studied because of their widespread availability and being easy to care for. So I'm going to start. My first slide on ciliates is conditioning in paramecia. And several notable studies have described how paramecia can be classically conditioned. The paper I'm going to bring up for this presentation is titled Classical Conditioning in Paramecia. I'll provide 3 interesting quotes from the paper. The first is the is indeed responsible for the rise in anticipatory CRRs and subject strain with paired CS and UCS is again reinforced by the performance of the second control group in this experiment, which went through 2 phases of training. The second quote is, thus, from the present experiment, it appears that paramecia are capable of discriminating 2 arbitrarily chosen vibratory stimuli, thus fulfilling part of Neil Miller's criteria for what he calls grade A certified learning. Third quote, which is probably the most interesting of the 3, is demonstration of learning in protozoa is hard to accept because it is inconsistent with the dominant assumption that learning is a property of synaptic interactions and not of the cells themselves. While the present study does not establish beyond a shadow of a doubt that protozoa are capable of learning, the burden of proof now lies with the contrary point of view. And I have included a link to the paper if anyone wants to look at it. It's very interesting. And to continue, a similar study titled Discrimination Learning in Paramecia stated in the abstract that previous attempts to condition a one-celled organism, the paramecium, by either classical or instrumental procedures have yielded equivocal results. The present experiments were designed to determine whether the use of positive reinforcement provided by DC electrical stimulation at the cathode, which had previously been shown to be attracted to paramecia, can be used to train these organisms in a discrimination learning task. The results indicate that such learning did take place. The abstract for the study, Paramecium Learning, New Insights and Modifications, states that our results indicated that in contrast to the previous reports, the paramecium can only associate higher light intensities with cathodal stimulation, and it cannot associate lower light intensities with cathodal stimulation. The conclusion, though, states in part that, in conclusion, our results in line with other studies suggest that paramecium can have associative learning. Meanwhile, there are several questions about learning in paramecium that need to be addressed, such as what is the exact molecular pathway that governs this behavior? Now I'm going to move on to the next ciliate, in decision-making in Stentor-Roselli. This is actually a pretty recent study. I only learned about this about maybe a month ago or a little over a month ago. The ciliate Stentor-Roselli is a species in the genus Stentor. An article from the website eurekalert.org describes a study done by Dartmouth and Harvard in which Stentor-Roselli displayed complex decision-making behavior. Toward the beginning of the article, it is stated that in 1906, American biologist Herbert Spencer Jennings reported that Stentor-Roselli exhibited complex behavior. In response to an irritating stimulus, Jennings said that Esforzelli engaged in 4 distinct behaviors. These were bending, ciliary alteration, contraction, and detachment. The news that the organism, which lacks a central nervous system, possessed sophisticated sensing and response mechanisms sent waves through the scientific community. The findings also played a key role in early scientific debates about animal behavior. The study ended up confirming Jennings' findings, with the article going on to say, our findings show that single cells can be much more sophisticated than we generally give them credit for. They also say they had to be clever at figuring out what to avoid, where to eat, and all the other things that organisms have to do to live. I think it's clear that they have complex ways of doing so. Now, I'm going to move on to bacteria. They need a little introduction, but I will provide a little information about them. They are prokaryotes, which means they don't have a nucleus. They have a nucleoid instead. And some do have flagella, which are used for mobility, which they are kind of similar to the cilia. They kind of have the same purpose. They're used for mobility and grabbing food, I believe. So yeah, that's the information on bacteria. There's a plethora of evidence that suggests bacteria are intelligent. An article from New Scientist titled "Why Microbes Are Smarter Than You Thought" each other, make decisions, form communities, and even accelerate mutations to gain new abilities. I'll provide 3 interesting quotes from the article. The first is, if the B subtilis individuals are growing in a food-poor area, they release chemicals into their surroundings. These essentially tell their neighbors there's not much food here, so clear off or we'll both starve. Bacteria talk to each other with chemicals. They do so for a host of reasons. Some of them are hard to understand unless you're another bacterium or dedicated bacteriologist, but one of the most straightforward is demonstrated by Bacillus subtilis. And the second is, many microbes can accelerate the rate at which their genes mutate. This allows them to obtain new abilities that may be helpful when conditions get tough. This is a risky strategy since many of the new mutations will be harmful or even fatal, and is in effect a last-ditch tactic when there's nothing left to lose or little left to lose. Similarly, some bacteria move according to the presence of chemicals in their environment. Behavior called chemotaxis. E. coli, for example, move like sharks on the trail of blood if a few molecules of food are dropped into their environment. Another group of bacteria align themselves to the Earth's magnetic field, allowing them to head directly north or south. Known as magnetotactic bacteria, their special ability comes from specialized organelles loaded with magnetic crystals. That is very interesting. And then I'm also going to go over another thing. It's sight in bacteria. This is something I've known about for a little while. I thought it was very interesting when I first read about it. Excuse me. I still think it's interesting. An article from Live Science titled "Bacterial Slime Acts as Teensy Eyeball" discusses how cyanobacteria, which is what I mentioned at the beginning of the video about how they're similar to their ancient ancestors. So this intricacy arose pretty early on, which seems unlikely in a naturalistic scenario. But they can see in a way similar to how people and animals see. To quote the article, slimy microbes called cyanobacteria use their teensy bodies as lenses to collect light and see before growing little legs to inch towards those rays. That means the basic inner workings of these miniature light collectors may not be so different from those of cameras or the human eye. Then one of the researchers says the idea that bacteria can see their world in basically the same way that we do is pretty exciting. And he goes on to say, our observation that bacteria are optical objects is pretty obvious with hindsight, but we never thought about it or thought of it until we saw it. And no one else noticed it before either, despite the fact that scientists have been looking at bacteria under microscopes for the last 340 years. So I did provide an illustration that came with it was, I believe the illustration was in the Life Science article, and it compares the camera, human eye, and the cyanobacteria eye. And this is another very interesting thing. I mean, it's not directly related to intelligence, but it is related to, sensory perception, so I want to include it. And now this is kind of similar to what happened in the paramecia, this conditioning of bacteria. An article from show bacteria can learn and plan ahead describes a study from the Weizmann Institute of Science in which bacteria were conditioned. Excuse me. The article begins by stating that bacteria can anticipate a future event and prepare for it. In a paper that appeared on June 17th in Nature, the researchers, they ended up examining microorganisms living in environments that change in predictable ways. Two other interesting quotes are E. coli, for instance, which normally cruise harmlessly down the digestive tract, encounter a number of different environments on their way. In particular, they find that one type of sugar, lactose, is invariably followed by a second sugar, maltose. They checked the bacterium's genetic response to lactose and found that in addition to the genes that enable it to digest lactose, the gene network for utilizing maltose was partially activated. When they switched the order of the sugars, giving the bacteria maltose first, there was no corresponding activation of lactose genes, implying that bacteria have naturally learned to get ready for a serving of maltose after a lactose appetizer. And the final quote is, to see whether the microorganisms were truly exhibiting a conditioned response, they devised a further test for the E. coli based on one of Pavlov's experiments. When Pavlov stopped giving the dogs food after ringing the bell, the conditioned response faded until they eventually ceased salivating at the sound. The scientists did something similar using bacteria grown by Dr. Erez Deckel in the lab in the cell biology department in an environment containing the first sugar, lactose, but not following it with maltose. After several months, the bacteria had evolved to stop activating their maltose genes, the taste of lactose, only turning them on when maltose is actually available. Of course, I believe the term evolved is a little bit incorrect, but of course, this is very good evidence for creation, so I'm just going to read the quote as it is, And that was it on bacteria, conditioning on bacteria. Now I'm going to talk about viruses. This will be the last of the three that I'm going to be discussing for the presentation. And like bacteria, viruses need a little introduction. But what's interesting and unique about them is that when compared to ciliates and bacteria, scientists don't even consider them to be viruses, to be true living organisms. They're specific also to regarding, what organisms they can infect. For instance, the bacteriophage on the right, which is actually a real photo of one, it almost looks robotic. They can only infect bacteria. So, like one of those couldn't infect a person, and the flu virus couldn't infect bacteria. So, all right, intelligence and viruses. Even though many scientists consider viruses to be non-living, they display remarkable decision-making capabilities similar to ciliates and bacteria. An article titled "Researcher Teases Out Secrets from Surprisingly Intelligent Viruses" describes the intricacies of viruses in detail. To quote at the beginning of the article, viruses are very intelligent. They can think, they do things that we do not expect, they adapt to the environment, they change themselves in order to survive, said Lay, professor of molecular microbiology and immunology and a Howard Hughes Medical Institute investigator. The article concludes by saying that after all these years, studying viruses' shifty ways continues to leave Lace with a sense of amazement. Part of this comes from the ability of viruses to shuffle genes with as much depth as some genetic engineers. Viruses can pick up pieces of cellular genes or incorporate their genes into the cell's genome. That means that evolution occurs all the time in viruses. It's a very dynamic process. That's why I always feel that viruses are alive. And I also have the link to that. Also, I'm going to move on to decision making in viruses. An article from Georgia Tech describes how the viruses can communicate with each other so that they can actually decide if they want to kill the host or let it live, which is very pragmatic, if you think about it. And I think that it's the intelligence and the ciliates and bacteria; those are interesting, but with viruses, though, they are not thought of as even living organisms, and they still possess such an ability to adapt to, make decisions. It really is amazing. But to quote the article, what has confounded the virology community for some time is the observation that the cell fate of a bacteria infected by a single virus can be dramatically different than that infected by two viruses. And it goes on to say, our study suggests that viruses can collectively decide whether or not to kill a host, and the individual viruses talk to each other as a result of interactions between viral genomes and viral proteins that direct the infected host to produce. Excuse me. This second quote describes the study in more detail, stating that for this study, the researchers analyzed the decision circuit that determines whether a virus initially chooses the pathway that kills the host cell, called the lytic pathway, or the pathway where it remains dormant inside the host cell, called the lysogenic pathway. When the lytic pathway is selected, the virus utilizes bacterial resources to replicate and then destroys the host cell, releasing new viruses that can infect other cells. In contrast, in the lysogenic pathway, the viral genome inserts itself into the bacterial genome and replicates along with it, while repressing viral genes that lead to lysis. The virus remains dormant until host conditions change, which can result in a switch to the lytic pathway. And now, viral decision making continued. There's another interesting source. This article from Science Daily, it describes viral communication, saying that they may be stealthy invaders, but a study at the Weizmann Institute of Science reveals a new chatty side for some. For the first time, viruses have been found communicating with one another. This communication, short posts left for kin and descendants, helps the viruses reading them to decide how to proceed with the process of infection. And the research was reported in Nature. There's a quote I included. At the beginning of infection, it makes sense for the viruses to go to the fast replication, kill the host route. But if they are too gung-ho, there won't be any hosts left for future generations of viruses to infect. At some point, they need to switch strategies and become dormant. Excuse me. The molecule we discovered enables each generation of viruses to communicate with successive generations by adding concentration of the arbitrum molecule. Each virus can then count how many previous viruses have succeeded in infecting host cells and thus decide which strategy is best at any point in time. The team found that the peptide binds a particular viral protein, which they identify as the genetic inhibitor of the dormancy life cycle. This binding activates the inhibitor, shifting the balance into choosing dormancy. So that was my slides on viruses. So to kind of conclude, I'd like to ask again, are these microorganisms intelligent? Before I go any further, I do want to point out, though, that I'm not suggesting that they have the same type of intelligence that animals or humans do. Indeed, I'm suggesting that their intelligence is more similar to that of computers or maybe robots. But based on the available evidence, they do seem to display some level of built-in knowledge on how to deal with certain situations, including management and problem solving. They're also highly adaptable as well. So considering the definitions listed at the beginning of the presentation, I think it's safe to say that they are indeed intelligent. I do believe that the evidence does indicate that there is some, built-in intelligence there, and there's this degree of adaptability, and that's really what is so amazing about all this. And it's really, it's amazing, that they can learn, they can adapt, they can be conditioned, or classically conditioned, like animals or people could be. I really just think it's just fascinating stuff. And of course, I'm going to end with a good Bible verse. Worthy are you, our Lord and our God, to receive glory and honor and power, for you created all things, and because of your will they existed and were created, which is Revelation 4:11 in ASB. So I have stopped sharing. I'm hoping that my mic didn't cut off any during the presentation, of course. I didn't want it to screw up any. I know that I just had some technical difficulties, but I'm hoping that everything worked out smoothly. But that was my presentation on intelligence and microorganisms. I really think it's just a very fascinating subject, and I really think it's something that more research needs to be put into it, to see exactly how they have these sophisticated, mechanisms for learning and adapting. I just believe it's very just amazing testament to God's glory. It makes me think of Romans 1:20, where it says that He is understood and seen through what He has made, so that we are without excuse. And it's pretty apparent, that He can be seen through what He's made, especially in viruses, bacteria, and other microorganisms, because He has left His fingerprint, I guess you could say, on the everything that He's made. But yeah, I may try to make another video like this at some point. I may make a follow-up video discussing the earlier point I made in the beginning of the video, because I'm not sure if I really went in depth enough in it. I probably did, but I keep thinking maybe I left something out. Maybe it could deserve a video on its own. But to reiterate it, I do believe that, because the cyanobacteria are so similar to their ancient ancestors, it just seems unlikely that all this intricacy could have appeared so quickly on in life's history. It's basically a very good example of stasis, which is, stasis is found all throughout the fossil record. I just don't believe that. I question whether or not, even if I was not a Christian, I would be skeptical of natural selection's ability to create. I know that some secular scientists are also skeptical of that, or have been, especially Lynn Margulis, who had commented that natural selection maintains but it doesn't create. And I don't think that mutations would really, random mutation, natural selection, I don't think that would, considering the small amount of time and all that arose, it just seems unlikely. And I think that it's pretty apparent that these organisms were designed, they were created by God, to reproduce according to their kinds, just like everything else. But yeah, that pretty much wraps up everything. I will probably, hopefully, make another video maybe within a few weeks. I'm going to just see what ideas I get. I might research the topic of what I just presented some more, and I'll maybe just make a follow-up video. But yeah, I enjoyed this presentation. I enjoyed making this video, despite the technical difficulties. I was hoping everything went smoothly while I was sharing my screen, because I couldn't tell if I was actually talking during that time. But yeah, hopefully I will make another video soon. So thanks for tuning in, guys, and God bless.